Hello everyone, welcome to my YouTube channel. This is again Sir Steven, and a part of this microbiology and parasitology lecture series, I will be presenting to you the cell structure and function of your bacteria and archaea. But before we go further into the lesson proper, I will be sharing with you a case study which is very interesting to know. This is all about kidney infection that leads to mental confusion. And the case study goes like this. Pete is a 68-year-old retired engineer. He remains mentally and physically active and is generally in good health. That's part of the reason he was so surprised that a couple of weeks ago when he found blood in his urine. Pete went to his doctor and he was diagnosed with a kidney infection. The doctor prescribed a 14-day course of antibiotics. After this first week or so, Pete started to feel better, so he stopped taking the antibiotics. About 2 days ago, he noticed that the pain in his back had returned. He also felt a little dizzy and seemed to get winded more quickly than usual. Pete thinks he should go back to the doctor, but wants to wait until after his granddaughter Sarah visits. She doesn't get a chance to visit very often, and they have a great planned day, a picnic in the park and nice long walk along the lakeshore. Pete is thrilled when Sarah arrives, but Sarah looks worried when she sees her grandpa. He's quite flushed and is a little bit sweaty and seems to be a little bit confused or disoriented. Although Pete insists that he is fine, Sarah really wants to take him to the emergency room. Pete continues to argue, but Sarah swears that she won't have a bit of fun if she's worried about her grandpa all day long. Finally, Pete agrees to go to the ER. And there is a question that is the starting point of inquiry to all of this. The question goes like this. Is Pete's earlier kidney infection connected to his current symptoms? What do you think? We will be answering that one during our class discussion. And before I introduce to you what are the subcellular components of your archaeans and bacteria, let me introduce to you the microbiology, which deals with particularly the small living things. It's a study of the small, minute microorganisms. And if we talk about microbiology, we'll be answering a question: what does a living thing mean, and how do we define life? Scientists once thought that living things they're composed of special organic chemicals such as your glucose, amino acids, and these molecules carry a life force which is found only in living organisms. These organic chemicals were thought to be formed only by living things and to be very different from the inorganic chemicals of your non-living things. And again, biologists agree that all living things share at least 4 processes of life. That should be growth, reproduction, responsiveness, and metabolism. There is a small free-living microbe. We call it the bacterium mycoplasma. It is non-motile. Why is it alive even though it cannot move? Why do you think so? Furthermore, this is the characteristics of life and their distribution in microbes. So we have an example of 5 characteristics, and we compare it within the microbes. One group of bacteria, archaeans, and the eukaryotes, and another group of the acellular microbes, we call them the viruses. So if it has growth, it should be increasing in size. It happens in all the bacteria, archaean, and eukaryotes, and it does not happen in your viruses. Increase in number It happens in all microbes except for the viruses, where the host cell will replicate the virus. Responsiveness. It occurs in all microbes except for the viruses, where reaction to host cells is seen in some viruses. Metabolism. It happens in all microbes, but the viruses uses your host cell's metabolism. And lastly, for cellular structures, these were the membrane-bound structure which is capable of all above functions. For bacteria, archaeans, and eukaryotes, it is present. However, in the viruses, it lacks the cytoplasmic membrane or your cellular structure. Moving on, we will talk about the overview of your prokaryotic and eukaryotic cells. In the 1800s, two German biologists, Theodor Schwann and Matthias Claden, developed the theory that all living things are composed of cells. And they also take a note that cells are living entities. They are surrounded by a membrane that are capable of growing, reproducing, responding, and metabolizing some of the characteristics of life, or the processes of life. The smallest living things are single-celled microorganisms. Looking at this micrograph, in letter A, this is taken by using a scanning electron microscope, and letter B, it was taken from a light microscope. So these are an example of the cell types. Okay, in letter A, you notice this is a bacteria, okay, named Escherichia coli. And letter B, this is a eukaryote, and you notice the presence of these hair-like structures surrounding this unicellular organism. This is a eukaryote, and we will take note of the bar scales on the lower right portion of these micrographs. They're being measured using the micrometer. And this is an example of an artist's sketch of your prokaryotic cells. Prokaryotic cells include bacteria and your archaeans, and the artist has extended an electron micrograph to show the three dimensions. Note that all prokaryotic cells contain all of these features. It might be present in bacteria but not in archaea, and vice versa. We have the ribosome, cytoplasm, nucleoid, which is the counterpart of your nucleus in your eukaryotes, glycocalyx, your cell wall, cytoplasmic membranes, flagellum, and inclusions, and this were being taken from your transmission electron microscope, your TEM, and the Bohr scale reads 0.5 micrometer. Moving on, in 1985, an Israeli scientist discovered the single-celled microbe Epilopicium fischelsoni. This organism is so big that it's visible with your naked eyes. The question is, why did the scientists initially think that Epilopicium was eukaryotic? And what discovery revealed that the microbe is really a giant bacterium? The photo in the background is one of the hosts of the single-celled microbe E. fischelsoni. This is your Candidatus epilopicium, which is a genus of a gram-positive bacteria which forms a symbiotic relationship with the fish, or the surgeonfish, in the background. These bacteria are known for their unusually large size. Many is ranging from 200 to 700 micrometer in length. So you have a clue that your Epilopicium fischelsoni is a gram-positive bacteria? So you have to answer these following questions. Furthermore, this is an artist's sketch of a typical eukaryotic cell. The one you're seeing on this portion is a micrograph from a TEM, and to tell you that not all the eukaryotic cells have all these features. Again, the artist has extended the electron micrograph to show the 3 dimensions. There is also a difference in magnification between this cell and the prokaryotic cell from the previous figure. Okay? The question is, besides the size, what major difference between prokaryotes and eukaryotes was visible to early microscopists? What do you think? Yes, you're right. The eukaryotic cells contain nuclei, which are very visible with light microscopes, whereas prokaryotes lack. In the laboratory, you have been exposed to your cheek cells being stained with methylene blue, and one distinguishing feature that it's a eukaryotic cell is the presence of, again, a stained spherical structure which can be seen in the microscope, and those were your kernel or your nucleus. And these are the external structures of your bacterial cells. Many cells have very special external features that enable them to respond to other cells and their environment. In bacteria, these features include your glycocalyces, flagella, fimbriae, and pili. This is a photo or an artist's sketch of a chicken egg which measures 4.7 centimeter in diameter, an equivalent to 447,000 micrometer. Okay, this asterisk will tell us that actually the insert box on the egg would be too small to be visible. The width of the box would be about 0.002 millimeter, and these are the approximate size of various types of cells. Again, the photo on your left is a bird's egg, which could be the largest cells. Okay, note that your staphylococcus, a bacterium, is smaller than Giardia, which is a parasitic protozoan. This bacterium here is smaller, and if you take a look at a virus, a smallpox virus, which is the orthopox virus, this is very small as compared to your staphylococcus. It's 0.3 micrometer in diameter. Okay, again, viruses are not cellular organisms. Another portion here of a micrograph comparing a glycocalyx and its capsule and the slime layer. These were both taken from your transmission electron microscopes. This is a figure from an artist's sketch of a proximal structure of your bacterial flagella. You will see here a detail of the flagellar structure of a gram-positive cell. Okay. Again, what is the purpose of your flagella? Of your flagellum? Yeah, they help in locomotion. And in letter B, you will also see the detailed flagellum of a gram-negative bacterium. And there's a difference, okay, between the flagella of a gram-positive bacteria from a gram-negative bacteria. We have these filaments, they rotate, okay, during the runs. They also have this letter A, the Peptidoglycan cell wall, which is quite thick, and the cytoplasmic membrane. In the gram-negative bacteria, they are composed of they are multi-layered, okay, with their outer protein rings. They have the rods, integral proteins, inner protein rings, and another integral protein, which forms the basal body. And on the other side, we can see the cell wall, which is composed of an outer membrane and a peptidoglycan layer, which is very different from your gram-positive bacterium. Another photo, micrographs of the basic arrangements of your bacterial flagella. When you say peritrichus, it's all over, okay? All over the body of the bacterium. When if it's single polar flagellum, the flagellum is situated on the either side of your rod-shaped bacteria. And this one is a tuft of a polar flagella. It comprises one or more flagellum on the either side of your bacteria. This is an eggshell filament, okay? In letter A, it will show you a scanning electron micrograph of two spirochete cells of this microorganism called Borrelia burgdorferi. Okay, this B. burgdorferi is the causative agent of your Lyme disease. In letter B, this is a diagram of your eggshell filament, which is wrapping around a spirochete. And when you take a look at your cross section in this photo, it reveals that an eggshell filament is composed of your endoflagella. This is your eggshell filament, and this is the course of action of the spirochete cells of B. burgdorferi, which cause your Lyme disease. Okay, so this diagram here of your eggshell filament that is being wrapped around the spirochete, revealing the tumble run course of action of your bacterium. Okay, so when does the bacterium run? When the favorable environmental condition induce it runs. Okay, it always follows the attractant. Okay, are we clear with that? So moving on, this is another subcellular structure of your bacteria. We call it the fimbriae. Okay, this is the flagellum, which is the longer apparatus, and the smaller ones are called fimbriae. This is an example of a bacterium called Proteus vulgaris. Okay, it has both flagellum and fimbriae, coming from an SEM. Biofilms is taken from an SEM in the bar scale of 6 micrometer. These biofilms are also an aggregate of bacterial colonies that are situated inside the human body. They could be elsewhere. They could be in your skin. They could be transient. They could be the resident. Okay, these biofilms are organized layers of systems of microbes, which were being attached to any of the body's body surface. This is another. This is a synthetic environment coming from hospitals. So most of your nosocomial infections that are these biofilms from a medical tubing is forming, okay, and sometimes will cause the severity of the disease if it will disrupt the normal microbiota of humans or the living organism. This is an example of bacterium called Escherichia coli. Okay, and this pilus or pili is being shown in here. So this mode of action, okay, is connecting one Escherichia coli to another Escherichia coli. And the question is, how are pili different from bacterial flagella? Because they look like the same. From the previous slides, you also see that flagella is quite longer structures. There's a fimbriae, which is a hair-like structure. But right here, the pilus or the pili is resembling the flagellar structure. So how do they differ? Of course, bacterial flagella are flexible structures which helps the cell to rotate and propel. Whereas your pilus or pili, these are hollow tubes. Okay? They are used to transfer your DNA from one cell to another. These are also an artist's sketch of your bacterial shapes and arrangements. Okay? In letter A, you'll see a spherical cocci which may be single or in arrangements such as your chains. Okay? These chains or arrangement of your spherical cocci, we call it the streptococci. If it's forming in clusters, although not shown in here, the cluster, we call it the staphylococci. If it's here, this structure looks like a cuboidal packet, we call it the sarcini. Okay? In letter B, you'll show here a rod-shaped bacilli which may be arranged in single. This is on top, and it could be arranged in chains. These are the comparison of the structures of glucose, your NAG or the N-acetylglucosamine and your N-acetylmuramic acid. Okay? They are linked together in your peptidoglycans, and the blue shading here, you can see that it's indicating the difference between the glucose and the other two sugars. This is the glucose and these are the other two sugars which are linked together. Okay? The orange boxes will differentiate NAG from NAM. Okay? It highlights the difference between these two components of your bacterial cell. This is a possible structure of a peptidoglycan. Again, your peptidoglycan is a part of your bacterial cell, prokaryotic cell, and they're composed of chains of NAG and NAM, and all together they are linked with your tetrapeptide cross bridges, and in some cases they are connecting chains of amino acids, and they're forming a tough yet flexible structures. The amino acids here, the connecting amino acids of the cross bridges, will differ amongst your bacterial species. Another photograph from the comparison of your cell wall found in your gram-positive and your gram-negative. In letter A, you will see a gram-positive cell wall. Again, it has a thick layer of peptidoglycan and lipoteichoic acids. The purpose of it is it anchors the wall to the cytoplasmic membrane. Again, cell wall is attaching to the cytoplasmic membrane using your this vertical structures here. We call it the lipoteichoic acid. Next thing is your gram-negative cell wall. Although it has a thin layer of your peptidoglycan and the presence of your outer membrane, it's composed of your lipopolysaccharides. Okay, it contains the lipid A. It also have a phospholipid bilayer and proteins. Okay, and interestingly, your lipopolysaccharide has lipid A. What's the purpose of your lipid A in human physiology? Very good. Lipid A can cause shock, blood clotting, and fever in humans. Another note to consider is there are bacteria without cell walls. Okay, a few bacteria such as your Mycoplasma pneumoniae lack cell walls entirely. In the past, these bacteria were often mistaken for viruses because of their small size and the lack of cell walls. However, they have other features of prokaryotic cells, such as your prokaryotic ribosomes. You'll see the photo at the background. These are your Mycoplasma pneumoniae. We also have cytoplasmic membranes. Okay, this is where your subcellular organelles in eukaryotes were floating, and this gel-like substance, we call it cytosol, is holding everything, all of these organelles in your cells. Okay, whether it be membrane bound, such as in eukaryotes, and non-membrane bound, such as in prokaryotes. And beneath the glycocalyx and the cell wall is, again, we call it the cytoplasmic membrane. They may also be referred to as your cell membrane or a plasma membrane. You'll see in the background photo is a phospholipid bilayer. It has a hydrophilic heads and hydrophobic tails, because these hydrophobic tails are composed of fatty acids. Again, your head is hydrophilic, it loves water, and the tail is hydrophobic. And beneath the surface of your phospholipid bilayer, there are embedded integral proteins. Okay, they are transcending in the sophisticated layer of your cell membrane. They have integral proteins here, which are transcending in your cell membrane, and there are also peripheral proteins that are located on the either side. Whether it could be, this one is located within the cell, because it's here is the cytoplasm, and sometimes it could be located outside the cell. Okay, so this is the structure of a prokaryotic cytoplasmic membrane. We also have to consider these electrical potentials of the cytoplasmic membrane. Okay, the electrical potential in this case is negative 70 millivolts, and it exists in across a membrane because there are more negative charges inside the cell than outside it. So using this integral protein, okay, this the presence of your sodium ions and chlorine ions will pass through this integral protein. And as much as possible, we wanted to let the inside of your cell, or the interior of your cell, or the cytoplasm, to become more negative as compared to the extracellular fluid, which is more positive. This is also to maintain homeostasis. That's why they're doing this. The integral proteins will allow the passage of these ions. Moving forward, we'll take a look at this clinical case study. This is different from the first case study. A college sophomore, Nadia, is a star point guard for her school's basketball team. She's excited about the division's finals Friday night. She's even heard rumors that a professional scout will be in the stands. On Thursday morning, she wakes up with a sore throat. Her forehead doesn't feel warm, so she forces herself to attend her Thursday classes. But when she wakes up on Friday morning, her throat is noticeably worse. She still forces herself to attend the Friday morning class, but feels tired and much worse by noon. It is downright painful to swallow, and she skips lunch. Near crying, she heads back to the dorm and checks her temperature. Despite desperate, she walks to the student health center, where a nurse practitioner notices white spots on the back of Nadia's throat and on her tonsils. The divisional basketball game starts in 6 hours, but it takes only a few minutes for the nurse practitioner to perform a rapid streptococcal antigen test and determine that Nadia has a streptococcal pharyngitis, also known as strep throat. She will miss the big game. Strep throat is caused by an encapsulated gram-positive bacterium, Streptococcus pyogenes. And the only good news thing is that by taking the prescribed penicillin, Nadia should be ready for her next game, hopefully the quarterfinals. Questions like this: how does the capsule of Streptococcus contribute to the bacterium's ability to cause disease? What bacterial structures besides the capsule may be allowing Streptococcus to infect Nadia's throat? Penicillin works by interrupting the formation of Peptidoglycan. What bacterial structure contains the Peptidoglycan? And in a gram-positive organism, such as your Streptococcus, in this structure, is this structure typically thicker or thinner than it would be in a gram-negative bacterium? This is very easy questions, and you can ponder upon it while we are coursing through this lesson. This artist's sketch will show you some of the passive processes of movement across your cytoplasmic membrane. Letter A, this is very easy to transcend, okay? Your diffusion of your small or lipid-soluble chemicals through the membrane. It's very easy. Letter B, facilitated diffusion, okay? This is composed of several types of chemicals through a non-specific channel. Letter C, this is facilitated diffusion. This is through a permease of a specific chemical. So this is, it only allows all of these green triangles to go inside the cell. And letter B, this is allowing a lot, or there are three varieties of shapes and sizes of ions or molecules that are coming into the cell. Letter D is your osmosis, which is the movement of water through a specific channel or through the membrane. This osmotic regulation is also being facilitated by the cell membrane, and we have these things called aquaporids that allows the entry or passage of water into the cell. This osmosis, okay, of also known as the diffusion of water across a semi-permeable membrane. Letter A, you will see a membrane that separates two solutions of different concentrations in a U-shaped tube. Take note that this membrane is permeable to water but not permeable to solute. Okay, and after time has passed, this water has moved down into its concentration gradient until water pressure prevented the osmosis of any additional water. So this one gets it's like preventing, okay, the increase of water in this size, because again the solutes couldn't pass through except for water. So which side of the living tube more closely represents a living cell? Is it the left or the right? Left hand side or the right hand side? Here or here? And this is a mirror. Very good. This is the right hand side. Okay? It represents the cell because cells are typically hypertonic to their environment. And when you take a look at the hypertonic solution, these are, letter A, we call it the cells in isotonic solution, which experience no movement of water because it's being equalized. Okay? This is a cell without a wall, such as your animals and mycoplasmas. These are cells with walls, such as your plants, fungal, and bacterial cells. In hypertonic solutions, your cells will shrink because of the movement of water out of the cells. In letter C, hypotonic solutions, they will undergo the bursting of your cell because the water will come in. to the cell: isotonic, hypertonic, and hypotonic. From the previous slides, we've learned the passive transport. It doesn't require energy. But now, we are requiring energy in the form of adenosine triphosphate, or your ATP. This is the energy currency of your cell. And we have here uniport, which is the transport of your cellular energy, okay? And its chemical, and these chemicals were moving against its electrochemical gradient. So this chemical here is moving using these proteins, okay, via the use of ATP. Your ATP is dephosphorylated. That's why it became ADP, adenosine diphosphate. Letter B, it's called antiport because it the mechanism goes like, in an exchange of one spherical molecule, there should be a square or cuboidal molecule that will pass through. And in addition, your letter C is, we call it the coupled transport. Okay, this coupled transport includes two integral proteins that are used to make the passageway or the entry of these molecules, and it combines the two types of active transport, the uniport and the antiport, okay, or also we call this the symport. So this uniport coupled with symport, in this example, the membrane again is using ATP energy to pump one substance out through a uniport, and as this substance flows back into the cell, it brings another substance with it through the symport, and that's typically how it goes. Okay, what is the usual source of energy for active transport? Bet you know it. Now, these are the groups of translocation. It is the process involving a chemical change to the substance being transported. In this case, we add the phosphate. Okay, this is a 1, 2, 3, 4, 5, 6 carbon sugar, and attached to a phosphate, making it translocated. Okay, it changed the chemical composition of your molecule. Now, this is a table which incorporates the summary of the transport processes across your bacterial cytoplasmic membranes. We have passive transport processes, diffusion, such as oxygen, carbon dioxide, lipid-soluble chemicals. For facilitated diffusion, we have the glucose, fructose, urea, and some vitamins. For osmosis, it's only water. For active transport processes, in active transport, we have your sodium ion, potassium, calcium, hydrogen, and chlorine. And for group translocation, most likely they are the carbohydrates, such as your 6-carbon sugar like a glucose, your mannose, and your fructose. This is an example of the granules of a polyhydroxybutyrate in the bacterium Azotobacter cocci. What is the function of a polyhydroxybutyrate? And this is a step of the endospore formation. Well, in bacteria, you can see this endospores forming, okay, especially in some dairy products, okay. So you notice that your milk, even though if it's refrigerated and kept in a very low temperature, but if it's not properly pasteurized, if heat wasn't able to kill all of these endospores, found in your dairy products, especially if the liquid ones, the milk, it might cause a spoilage. All right. And in the formation of endospore, the first step is DNA replication. Of course, they're replicated from the daughter strands from the parental strands. And we call it the vegetative cell. And forming a cytoplasmic membrane which invaginates to form a forespore. So from one vegetative cell, it forms two using the forespores. And for third step, the cytoplasmic membrane grows and engulfs a forespore within a second membrane. So there is a second membrane that is forming, okay? And the vegetative cell's DNA will disintegrate. So this endospore formation typically happens, okay, over a period of 8 to 10 hours. So let's take a look at the next step. So after the DNA of your vegetative cell will disintegrate, a cortex of peptidoglycan is deposited in between the membrane. So we call it the cortex, okay? Meanwhile, the picolinic acid and calcium ions will accumulate within the center of the endospore, thus forming the spore coat here. So once the endospore will mature, the spore coat will complete and it will increase its resistance to heat and chemicals by unknown processes. So this is making the bacteria more resistant to any physical agent or chemical agent. And the spore is now being released from the original cell. And you can also check the formation of endospores. For instance, your botulinum toxins, most especially when you can see a canned good which has dents, okay? Although it's properly sealed, but the tendency of your botulinum toxins, they will be releasing these endospores and they will releasing as alongside with it, the toxins will be accumulated within these canned goods, and when the person will or humans will consume these canned goods, eventually they will become poisoned with the toxins of your botulinum bacteria. Okay, so this is an endospore formation process. This is another photo of a light microscope of a simple helical cytoskeleton. This is a rod-shaped bacterium, Bacillus subtilis, which has a helical skeleton composed of only a single protein. They are being stained with a fluorescent dye. Moving forward, this is another domain of life that we're talking about, and we'll discuss its major difference or comparing and contrasting it with the previous domain, bacteria. This is the external structures of your archaea. Archaeal cells have external structures which is more similar to those seen in bacteria, although they are both prokaryotes. We include glycocalyces, flagella, and fimbriae. Some archaea have another kind of proteinaceous appendage called a humus, and we consider each of these in order, beginning with the outermost structures. We call it the glycocalyx. This is your humus, okay? We take a look at this structure. It has a grappling hook and a prickle. It's quite unique, okay? Since this domain of life is living in a very extreme and very harsh environments, Archaeans take a lead in these evolutionary forces that has formed their external structures. Also, with the representative shapes of your archaea, okay, we have letter A. This is a cocci, Pyrococcus furiosus. they are being attached to a rod-shaped methanopyrus candelaria. They are being attached to another archaeans, okay? A rod-shaped archaeans. Letter B is a quite different shape. It's irregularly shaped Archaean, thermoplasma acidophilum. So from the name itself, thermoplasma, it means to say it lives in a very hostile temperature and acidic lover. Okay, it's acidophilum, so it loves acidic environments and extreme temperatures. Letter C is another shape which is not being seen in the domain bacteria. We have here a square archaean, halo quadratum vals B. Okay, so what are these? What do you notice about this string-like extensions of your pyrococcus? What are they? Very good. We'll take a look at some structural characteristics comparing the archaeans and bacteria. Okay? And what are they made of? For glycocalyx, they are both a polypeptide or a polysaccharide. For flagella, they are present in both, and it depends on the diameter. Okay? Bacteria has a bigger in diameter. Okay, it grows in the tip. Both could be counterclockwise. In your bacteria, it could rotate independently clockwise to cause tumbles. You have seen the figure from the earlier slides. For fimbriae, they're both proteinaceous. In bacteria, they are gliding motility and both are helping in the biofilm formation. Pili in Archaea, none, present in bacteria. Hami is present in some Archaea, and they're being used for attachment, not found in bacteria. Cell walls present in the most Archaea and bacteria. C membrane, they are present in all. Okay. The difference are the linkages. For the archaeans, they have ether linkage, and for bacteria, they have ester linkage. And the layers for the cytoplasmic membrane in archaea, they have single lipid layer. It's kind of monolipid. But in bacteria, they are bilayered. Cytoplasm, we have a circular DNA for archaeans and 70S ribosomes. And ribosomal proteins are very similar to eukaryotic ribosomal proteins, which in fact tells us that a lot of research and in comparison with the genome of your archaeans, these cells were closely resembling the domain eukarya as compared to bacteria, even though they are both prokaryotic cells. In bacteria, they have bacterial proteins, the same thing with 70S ribosomes. For this engulfing of substances, the archaea are being surrounded with pseudopods, okay, and brought these materials into the cell. Bacteria, viruses, age and dead cells, these liquid nutrients are found in the extracellular solutions. And for exocytosis, the release of substances, in the archaea, they have these vesicle-containing substances that are found in the cytoplasmic membrane and dumping contents to the outside. Whereas in bacteria, the waste and secretions are being moved from inside to outside. And lastly, we take a look at the former case study that we're trying to solve. It's all about the Pete and his kidney infection. Does it lead to mental confusion? At the ER, Pete's vital signs are taken, his mental state is assessed, and samples of blood and urine are collected. Pete has elevated heart and respiratory rates. and his blood pressure is low. Blood work reveals that elevated white blood cells is a sign of infection and a presence of Escherichia coli, which is also detected in the urine sample. Escherichia coli has its pathogenic strains. Europe is a uropathogenic bacterium, which is also being found in some of the patients which has recurrent UTI and bladder infections, also with gastrointestinal infections, because they might be transferring from digestive system to the urinary system. Pete is admitted to the hospital and started on intravenous antibiotics. The doctor explains that Pete stopped taking his antibiotics before the original infection was fully under control. This allowed E. coli to continue growing and move out of his urinary system, bladder, kidneys into his bloodstream. He explains to Sarah that her grandpa is so sick because he is having a systemic inflammatory response to the Escherichia coli. The doctor also notes that it was good she insisted on bringing Pete into the hospital. One day longer and Pete could have been suffering from organ failure. Pete stays in the hospital for 2 weeks when he is discharged, and he is still a bit weak but recovering. The doctor continues Pete on oral antibiotics for another 2 weeks. Ultimately, Pete makes a full recovery. To end, these are the following questions that we need to answer for the purposes of discussion in this particular lesson. E. coli is a gram-negative bacterium. What cellular component is most likely to be causing Pete's systemic inflammation? Pete's current situation developed from his original kidney infection, which probably started as a UTI. What cellular structures would help E. coli establish an infection in the urethra and then move into the bladder, kidneys, and bloodstream? And this is the end of the slide. Although I couldn't give you the full-length details of this lecture series, you have to become familiarized with the functions of each of the organelles between archaeans and the bacteria domains of life. So up next, this is microbial metabolism, and I thank you so much for listening to today's lesson. I hope you will subscribe and like my YouTube channel. Have a wonderful day and stay safe.